So we have two very interesting talks today. The first from Jelena, and then the second one is from Nirupama. So how this is going to work is we'll have the presentation from Jelena first, and then we'll have a maybe 3 to 5 minutes time for questions. And then we'll go into the second presentation from Nirupama and another time for questions. And then at the end, if we have enough time and there are people who are interested, we can go back and we can ask questions to both speakers about their projects. We have Nirupama Saini, who is a PhD researcher in the Indian Institute of Science Education and Research in Kolkata, India. She's working on understanding the potential role of microplastics in affecting carbonate chemistry of coastal waters. So she was going to present live. So if Nirupama, if you can present, share your screen, and then we will go into the second presentation.
Okay, right. So I hope it is visible to all. Is it visible?
I can see it, it looks good.
Okay, thanks a lot. Hi everyone, I'm Nirupama Saini and I'm a PhD researcher working in Indian Institute of Science, Education and Research, Kolkata. And I'm working on understanding the carbon chemistry of coastal waters and how the microplastic is affecting the carbonate chemistry by affecting the microbial community of the ecosystem. So today I'll be talking about investigating the seasonal abundance and distribution pattern of microplastics in a mangrove ecosystem of northeastern coastal Bay of Bengal. So I'll start with a bit of introduction. So whenever we hear the word marine plastic debris, that the first thing which strikes our brain is the entanglement of marine animals caused by the plastic debris or the fishing gears. These fishing gears, entanglement of marine animals led to the huge loss of marine ecosystem. As we all are aware of the impact of this bigger plastic materials on the marine animals, but there is also hidden enemy working behind the scene, and which are the microplastics. Microplastic are the plastic particles which having a size lower than the 5 mm. So these are some images that is taken by our research group from the site. So moving on, when we talk about mangroves, the importance of mangrove has been globally acknowledged, in spite of the fact that mangroves continue to decline because of a lot of anthropogenic stresses they are facing. And plastic is one of that stresses that mangrove are facing. This is a global map overlay of mismanaged plastic waste and the global distribution of mangroves across the latitudes and longitudes. This map clearly illustrates that mismanagement of plastic waste is high in the areas where mangrove are still abundant. So this co-occurrence of mismanaged plastic waste and high mangrove abundance is going to be problematic because due to the root structure of the mangrove that decrease the flow rate of the water, lot of plastic debris and particles get deposit on the mangrove flow. So as you can see that in Southeast Asia, where the mangrove declining is at high rate, the mismanagement plastic waste is higher. As I mapped my study area also with the circle, you can see it on the screen. So actually the impact of this plastic waste on the mangrove has been poorly understood till date. So another question comes that why do we really need to bother about plastic? Why do we really need to study the plastic impact on ecosystem? So it's often forgotten that the plastic is actually the fossil carbon that is locked on the polymer form and it's circulating in the ecosystem. And roughly 7 gigaton of the fossil carbon is now in the form of plastic. And this plastic provides a niche to the microbial community, and a lot of organic carbons leaches out from this plastic waste that might be toxic and that also impact the ecosystem. And not only this, the microplastic, I talk about the microplastic, the lot of filter feeders confuse the microplastic with food particles, and that end up entering of the microplastic in the food chain. And a lot of polymers releases greenhouse gases when they exposed to the UV. So these all factors actually contribute is impacting the carbon cycle of the ecosystem. So talking about Sundaman, which is the largest contiguous mangrove delta mangrove forest, it's spread across the area of 10,000 kilometers square and is shared between India and Bangladesh. So my study area is located in Indian Sundarbans, which is located on the Ganga-Brahmaputra Meghna Delta. If I talk about Ganga, Ganga is the second largest contributor of the plastic to the ocean as per the global modeling system. So we can say that this area is going to be hugely impacted by the plastic pollution, as Ganga is going to carry a lot of the plastic to this area. Not only the high amount of fresh water discharge that this area receives, this area is also impacted by the saline water inflow from the Bay of Bengal. My study site is located on the Saga Island of the Indian Sundarban, which is the largest island of this Indian Sundarbans. And the study site is comprised of 3 stations, Station 1, Station 2, and Station 3. Station 1 is located inside a creek known as Chimaguri Creek, and which is a shallow water column. And Station 2 is located at the mouth of the Chimaguri Creek. And Station 3 is located in the Muringanga estuaries. As you can clearly see from this map, that a lot of human habitation is near to this area and aquaculture farms are there. So not only the plastic debris, which is being carried by the riverine discharge to this area, the local anthropogenic stresses is also going to impact this area. So I've been sampling from all the 3 stations on the monthly basis, collecting the surface water to calculate the microplastic abundance and to calculate the microplastic abundance in this area. So there are some pictures that I've taken from the site. Here you can see that on the right side of your screen that a lot of plastic debris is on the mangrove floor. So some studies has shown that this plastic, because they get easily entangled in the pneumatophores of the mangroves, they can cause the soothing of the plastic and cause the suffocation for the mangrove trees. And also there are, during my sampling time, I have encountered a lot of the nets and the fishing gears that got entangled into the mangrove roots and the stems, etc. So here are the results which I got from my sampling, started from the June 2020 to the study is ongoing, but I'm showing the data till late January of 2022. So here you can see that microplastic abundance ranges from 3 microplastic pieces per liter to 66 microplastic pieces per liter in all 3 stations. And it's the microplastic concentration is showing a huge trend. It's fluctuating in each month basis, on a monthly basis. This Station 1, Station 2, and Station 3 are the part of SBOTs, which I have mentioned on the slide. This is Sundaman Biological Observatory time series. This has been started in 2010. Till from that now, from these 3 sites, we are continuously monitoring the hydrological parameters till date. So to understand the seasonal impact on the microplastic abundance, I have plotted the box plot for monsoon and post-monsoon season to look at the microplastic trends. Here we can see that the concentration of microplastic is higher in a monsoon season, but it's lower in post-monsoon season. So it's obvious that in monsoon season, due to the high riverine discharge and also the local precipitation, that can cause a lot of plastics, a lot of the local plastics to get into the water column. The plastic is going to be higher, microplastic is going to be higher in the upper water column. And looking at the station wise, we can see that in monsoon, Station 2 is showing the high abundance of microplastic, and Station 3 is showing the lower abundance of microplastic. I can remind you that Station 3 was outside of the, it's outside in the Muringanga estuary, and Station 2 was located in the mouth of the Chimaguri Creek. But in post-monsoon season, station 3 shows a high abundance of microplastic as compared to the other two stations. This is the map, which this is the graph which I have prepared on the basis of morphological analysis of microplastics. So here we can see that for the fiber concentration is higher as compared to the other form of microplastic like fragment, foam, and microbead. And if we look at the graph on the seasonal basis, and we can see that not only the fiber, but fragment also shows a high abundance in monsoon season as compared to post-monsoon season. And looking at the microbeads and foam, this I have encountered very less as compared to the other forms of microplastics, and fiber constitute the major part of the microplastic on a morphological basis. So moving on, I also tried to understand the composition of microplastic on the basis of color. So why do we really need to understand the why it's required to understand the microplastic distribution on the basis of color? Because a lot of zooplanktons, the filter feeders, confuse microplastic with food because of the color. Like if there's green microplastic floating in the water, and some zooplankton end up eating it because they confuse it with a green algae. So this is important to understand the microplastic distribution on the color basis so that we can see the impact of microplastic on the ecosystem. So I've also classified in the seasonal basis so that you can understand how the microplastic color composition is changing with seasons. So on the right side, this is the fiber I have shown. So it's on overall, we can see that colorless microplastics are more abundant in monsoon as compared to the post-monsoon. However, I have encountered black, blue, brown, green, pink, red, and yellow colored microplastics there. And in the case of fragment, it's just opposite. Like in post-monsoon, I'm getting no colorless microplastic higher as compared to the monsoon season. So here I put some bright field microscopic images to the microplastic that I've encountered during my study. So the upper row, you can see some fragment pieces are there, and the lower, some fiber pieces are there, which I have encountered during my study. Moving on to understand more about the microplastic pollution in the water column, I have also collected the bigger plastic pieces during my collection of the microplastic samples to understand the local anthropogenic stresses going on the local area. So whatever the macroplastic or the bigger plastic fragments I collected, I classified it on the different bases. Like if we see from this pie chart, we can see that second most abundant macroplastic in that area is fishing gear. So it's giving an impact on us that lot of fishing activities happens in that area. So fishing gears includes ropes and fishing nets, and most abundant of with the 19% others and the plastic film. In others, I have got some face masks that we have got due to the COVID situations, and also some flowers and some fragments of small pieces were there were included in the others. And also plastic cutlery was there. This is due to the human habitation because and local human settlements that might be using this plastic cutlery that is ending up in that local environment. Knowing this, to understand the polymeric structure or the chemical composition of the microplastic that I have encountered in the area, I did chemical characterization using micro-Raman technique. And so I found out that polypropylene was in high abundance, which is one of the most abundant polymer type used in the packaging material. But here we are also getting the signal of polyamide, polyacrylamide, which is used in the formation of the nets or fishing gears. So what we can infer from this study, that microplastic concentration that I've already discussed range from 3 to 6 number of pieces per liter in the surface water of northeastern coastal Bay of Bengal. And we have already seen that on the morphological basis, microbeads and forms were in very low abundance, and fiber constitute about 60% of the total microplastic. And microplastic concentration was found to be high in monsoon as compared to post-monsoon season. If we talk about station-wise, then microplastic concentration is found to be high in station 2 in monsoon, but it was high in station 3 for post-monsoon. The abundance of microplastic was high in station 2 in monsoon. It could be due to because the station 2 was located at the mouth of the creek. And on the color basis, we can see that colorless microplastics were in high abundance. And the microRMN analysis or the spectroscopic analysis, we say that we encountered 14 different types of polymers with polypropylene as the most abundant type amongst them. So thank you all for listening to my presentation. And I put this last slide, that how I process my samples. If anybody interested can go through it. Thanks a lot. Now, please ask any question if there. Thank you so much.
Great, thanks, Nirupama. Do you mind? Oh, that's great. Okay, we have some questions coming in. The first was in the chat from Maria. She says, I think I missed it. Did you sample water or sediments? Is there a monsoon-related lag? I mean, after the monsoon season ends, I'd expect a resuspension of sorts. Is the color variability related to the characteristics of the plastic losing, in quotes, its color?
Hi, Maria. So thanks for the question. So I sampled the surface water sample. It was not the sediment sample. First question I have answered. Now going to your next question, which is there let me go through it once. Is there a monsoon related lag? I mean, after the monsoon season ends, I'd expect a resuspension of sorts in the color variability. So it could be the reason. It could be the one of the reason that plastic might be losing its color. So yes, it could be the reason. So I hope I haven't answered your question. Otherwise, you can just put it in the chat.
Okay. Barney has a hand up. If you want to, yeah.
Hello. Thank you. Can you hear me okay? Yes.
Yes.
So it looked like the time series that you showed for the different stations were cross-correlated. Did you actually look to see if there was a significant correlation between those three stations? And if so, what does that correlation tell you about the supply function? And it sure looked from the slide that you showed that there was a correlation. When you saw high values at station 1, you were also seeing high values at station 2. And why is that? Thanks.
Thank you, Barney, for your question. So let me answer it. So I have looked into the correlation, but if you go through the data very closely, then you can see that it's not correlating all the time. It's correlating for some months only, but sometimes station 2 shows higher, it goes to the higher value and station 2 comes back. But right now, because the study is going on, so I'm not doing the I'm not fully understand the correlation, but it's like there are somewhere there are correlation, but somewhere there is no correlation between the station. But yes, obviously that I'll look into it when I'll complete this time series work.
Thank you.
Thank you, Jelena, for your question.
Do we have any more questions for Nirupama or Jelena? You can talk about either presentation. No, well, if there's no, oh, no, that's a clapping hand. Or is that a raised hand for Kevin?
Oh, hi, can you hear me?
Hi. Yes.
I wanted to just jump in and ask a question about the microplastics presentation. Very good overview. I just wanted to know if you were planning to estimate loads of plastics down that system, and whether or not it's tidally influenced, and whether or not you know the direction of the concentration, the direction and amount of the concentrations that you're reporting, how that translates into total transport of those materials, in what direction, and if you plan to quantify that.
Hi, Kevin. Thanks for your question. So, yes, because I'm working on the 3 station, which is not very far, yes, but there's a distance between them. So we can understand the flow of the microplastics, but actually I'm working on how microplastic is gonna impact the carbon cycle and how it's changing the microbial community. So my main focus is on that. So I'll not be going that much deep, into depth of the effect of the tidal influences on the microplastics. I'll be looking at how microplastic will be affecting the carbon cycle or the microbial community of that ecosystem.
Thank you very much.
Thank you, Kevin, for your question.
Well, if there's nobody else, then I want to thank both presenters, Jelena and Nirupama, for great presentations this morning or afternoon or evening, wherever everyone is around the world. Amplifying Voices will continue, and we will be announcing the next round of speakers and times coming up shortly. So keep your eyes open for emails and posts on Twitter. And thank you all for coming and sharing time to hear from our great presenters. So thanks to everyone. I hope to see you all coming back next time.